my name is David Simpson. I'm a reader in the Wellcome-Wolfson Institute for Experimental Medicine here in Queens. My research is as part of a consortium, a UK-wide consortium called COG-UK, which has been sequencing the SARS-CoV-2 virus. And this has been an amazing experience working in this large collaborative network, which has actually produced more genome sequences than the rest of the world put together. So the purpose of this project is to sample sequences of the virus from right around the country, and then we can look at the variants that occur in these sequences and then study the spread and the transmission of the virus throughout the pandemic. So a genome is a genetic material that's required to encode all the information needed to make a virus. So inside the virus particle is some RNA, and this contains the code or the letters which have the information required to make the proteins that go up to make the virus. The sequences of each viral genome can vary slightly, so as the virus spreads, it accumulates mutations. So we have, in each genome, there's approximately 30,000 letters, and then just one or two of those may differ between different viruses. So we can put the sequences we get into the computer and compare those to the original strain in Wuhan. And in certain places we see just differences in one letter. And then by looking across the whole sequence, we can see individual viruses fall into different strains that share certain groups of variants. Being able to measure the individual variants between viruses that we isolate from patients is very useful, because we can follow the transmission of the virus. So if a virus has one particular mutation, we can then sequence the virus isolated from example, from patients in a hospital. And if they both have the same variant, we know this spread occurred within the hospital, whereas patients in the same hospital might have different variants, and then that tells us then that it must have been introduced separately into the hospital, and this allows the authorities to take the appropriate steps. So as I mentioned earlier, we can look at the variants that occur right across the genome of the virus, and what we see is that the viruses fall into certain groups that have similar sequences. So we can follow through from the initial Wuhan strain and see how different variants were introduced, and we can essentially draw what is like a family tree for the virus, and then we have different branches that share certain sequences, and these are called lineages, and we can track these lineages as they spread throughout the country and even globally, and see how the virus has been transmitted. So this allows us to follow particular strains of interest. So you're probably familiar with a strain that has been detected towards the end of last year, and then has become very prevalent over the Christmas period and now into the new year. So this is a strain that's been called B117 as the technical name, or otherwise known as the English or Kent strain, which has a very high transmissibility. And by sequencing various viruses from across the country, we can see how that strain has spread very rapidly. So this is one of our jobs, is to follow strains such as this, and then in particular there's been several strains that have similar characteristics to this English one, so they're not directly related, but they have some of the same mutations that we're concerned about. So we're at the moment looking out carefully to spot when those arise. But this project going forward then, one of our jobs will be to look out for these new strains of concern so that we can see where they occur and then the appropriate action can be taken. And the other thing that we'll be focusing on as we go forward is potential vaccine escape. So the development of the vaccine has been an amazing achievement, and it's been rolled out very rapidly, which is giving us real hope, and hopefully we'll conquer the disease. But one thing that's slightly of concern is that perhaps the virus will evolve to escape the effects of the vaccine. So what we want to do is monitor very carefully for that occurring. So if there is any individuals who've had the vaccine and then go on to get COVID again, we're going to sequence the genomes of those viruses to make sure that they're not evolving in such a way that's allowing them to escape the vaccine. But then to finish on a positive note, obviously this whole pandemic has been awful, but it has actually accelerated the rate at which we've done things, and it's catapulted genomic medicine maybe 10 years forward. And plans are already in place now that even when this pandemic is finished, we now have the groundwork and the infrastructure in place that we're going to have a dedicated genomic sequencing service in Northern Ireland as a direct result of this project going forward in the future.